Okay, thank you very much for the organization and especially for your perseverance to organize these conferences. And I really wanted to take the opportunity and to take the chance to talk about very speculative things or very crazy things or maybe crazy ideas or the kind of things. So I will not present you some really well done work, and I will just want to take the chance to discuss with mathematicians, statisticians, and biologists to try to answer or at least discuss about a very general question, that is, what can we infer from data, from biological data, and with some population biology models. And so I will just give you some thoughts and some very largely speculative thoughts. Okay, so just to give you a little background, so I work with mathematicians and probabilists, especially in a statistician for quite a long time now, as a theoretical biologist. And after many discussions with all those people, and I certainly missed some people, but after many discussions with probabilists and statisticians from everywhere, there is still very often there is one question is about what can we do with probabilistic models to get some information from data and to make some inference. And so I think this is one important question that is actually maybe new and relatively original because there are some new mathematical tools that are actually developed. So just to be clear about what I mean by inference, and especially in population biology, so I will focus on population biology here. So basically what we have in population biology is data. So we have some data, and from those data we show there are some patterns in this data, and what we want to know is, what we want to have some information of this from these patterns is, what are the processes that underlie the data that show these kind of patterns. And for all these kind of things, we need some models. So we need some models to acquire some data, for example, what kind of experimental design or what kind of observations I will make in natural population. How do I do the observations? So this needs some first kind of model. How do I extract information from the data to show some patterns? And this also needs some models. And how can I identify what are the processes behind this data and those patterns? And this needs also some models. So the models are really in the at the core of the problem of inference. And the idea, the question, the big question that we want to answer in population biology especially is, what are the information we can get about the world from data? So we have some data and we want to have some information about the world and about the population biology world. So this is a very, very old question in biology, and especially in population biology. And maybe this question is as old as the existence of data, of population data. So certainly more than 1 or 2 centuries. And it is still today a very important question. And there are many different papers and works that are trying to develop some new tools and new models to acquire this kind of information from data. So just 2 quick examples just to illustrate what we are trying to do. From data of genetic diversity, for example, so we can have some samples of genomic and genetic data from human populations all around the world, for example, here in Africa. And we want to know if from this genomic data, from this genomic diversity, we could reconstruct the demographic history of some populations. For example, here in Africa, and so this has been done, this has been reviewed recently in a very recent paper by Pereira and collaborators. And they showed that, for example, with some tools based on the estimation of divergence between haplotypes, based on coalescence, based on the coalescence theory, and some Bayesian computation theory, then it's possible to reconstruct some past demographic events. For example, that there has been some populations went out from Africa and then went back to Africa, and that there are some three or four different clusters of population which diverged some dozens, thousand years ago, something like this, and it depends on whether we are in the middle of Africa or in the eastern of Africa, and these kind of things. And here, this is very common to try to use genetic diversity to try to reconstruct demographic events and the demographic history of populations. And very often, the demographic models can be very complex. Here we can see that there are some different populations. The population can go out from Africa and then can go back to Africa. So we can see that the demographic can be very complex. And of course, population can interact, can exchange some genes at some rates, they can migrate or not, they can compete. So the underlying processes that produced this genetic diversity is certainly something very complex. Another example is from more about the community ecology discipline. And the question is, how can we explain the diversity that we observe in a given community? And this is one very famous example on a tree community, a forest tree community in the Barro Colorado Island, which is an island in the Panama Canal. And there have been many studies and observation from this tree community. So we know almost every tree in this community. We know its species, we know its age, we know its width, and so on and so on. And the kind of thing that we can do with this data is this ranked representation of the diversity of the tree. So here on this kind of graph, this kind of relationship, we have on the abscissa, we have the ranked based on the abundance of a species of tree. Okay, so basically from the left to the right, we have the most abundant to the less abundant tree in this community. And on the y-axis, we have the relative abundance of this species. And very often what we observe in this kind of community is this shape of the relationship between the rank of abundance of the species and the relative abundance of the species. So this is very common that we observe this kind of S shape of the relationship between the rank abundance and the relative abundance of the different species in this community. So the question is, how can we explain and what kind of information can we get from this relationship between the rank abundance and the relative abundance of the species in the community? And it's now a hot debate for more than 20 years. And basically, there are two kinds of ecologists that are debating about what are the most important processes underlying this shape. And there are some ecologists are saying that the most important processes are local extinction and immigration, so such a kind of neutral processes. And other ecologists think that interaction between species are the most important processes. And so there is a debate, a very hot debate, to try to disentangle from this data what are the main important processes underlying these patterns. Okay, but here once again, of course, the real community is something very complex, because even if we focus on the tree community, there are many of other species interacting with the trees. There are fungus, there are animals, birds, there are many other plants than the trees, and so on. So the community itself is much more complex than the only tree community that we focus on. So here again, it's very difficult to know and to disentangle all the different processes that really explain what the patterns we observe. So in fact, in population biology, there are many different limits to inference. So for example, I just give a list of few points here that of course, the models that we use to infer and to interpret the data are much simpler than natural populations, of course. If we want to interpret the pattern in the tree community in the Panama, for example, we cannot take into account all the spatial interaction or the different species that interact. We cannot take into account the environmental heterogeneity, and so on and so on and so on. So the models are much simpler than natural populations, of course. But even if we want to make, and sometimes we make some very different, some very complex models with many different parameters, and sometimes we want to do that, and especially people who are trying to infer some the demographic history of some population, for example, of human population, they tend to make the models more and more complex with more and more parameters. And so it comes the problem that how is it possible to estimate all these parameters? Is it possible to compare very different and complex models and these kinds of things? Very often, especially when we deal with very complex models, very often we can see that several processes or several different scenarios or several different parameters, hypotheses on parameters can give similar patterns. For example, it is well known that if we look at the genetic diversity on a genome, we know that the demographic history, demographic past of a population can give some patterns of genomic diversity, which is very similar to selection. Okay, so this is one of the problems we have, is that several different processes that are totally different in natural population can give similar patterns, and it's a really difficult, important question to see if it's possible to develop tools and models that are able to disentangle different processes that can give rise to similar patterns. Another problem, another issue, another limit for inference is that most often we don't have any clue about the past of the populations. It can happen that we have some fossil records or some archaeological data or these kind of things, but of course they are very, very limited. And most often, in most cases, we don't have any clue from the past, and especially we don't have any idea of the initial conditions of a given population, of course. And this, of course, it's a problem, because it really limits to what we can say and what we can infer from data if we don't know even the initial condition of a population. And maybe a more methodological limit is that most often we don't have access to independent data to validate the inference. For example, if you if you try to infer some demographic history of human population from Africa, then it would be great to have some independent data on which you could validate your interpretation from the African populations. But most of the time, this is not possible to validate the quality of inference from independent data, except of course from computer simulations with some based on a particular model. But of course, it gives some security problems because you validate your inference methods on the models you want to test. So it's in some kind some circular reasoning. And maybe a last limit is that we have more and more data, and especially genomic data, but big data does not necessarily mean much information. And sometimes it can give very little information because there are many autocorrelation between populations, for example, or within population, the different genomes tend to be very similar, or spatially distributed individuals tend to be more similar than others, and there are non-independence of data if we look at different traits, for example, then we can see that we can observe that sometimes there is no independence between the traits that we observe and that we measure. So big data does not necessarily means much information, and it can also be limits to inference nowadays. Okay, so what I wanted to do in this presentation is, okay, most of the time when we do some inference in population biology, we are very often interested in making some inference with very complex system, okay? So, for example, the African populations or the tree community in Panama and so on, these kind of things. So I wanted to make a twist on this and say, okay, let's start with maybe the most simple ecological system we can imagine, and let's say, okay, what kind of information can I get from the most ecological system? Let's try a very simple model, and let's try to see if it's possible to make some very good inference with very simple models in a very simple case, in a very simple ecological system. Okay, so what kind of very simple population system can we think of? Okay, so let's start with the basics. So maybe one of the simplest organisms we can imagine are bacteria. So we can imagine a population of bacteria. And why? Because bacteria are clonal, so there is no sexual reproduction. There is a relatively limited and controlled genetic exchange between individuals and so on. We can have very homogeneous populations from bacteria with all the same genotypes, and so all the same phenotypes, and they are relatively simple organisms. Bacteria are also actually it's possible to make some individual counts with microscopes. Okay, so we can, if we work on bacteria, then we can have some almost exact dynamics data, because we are able today to count individual cells in an experiment. If we work in a controlled environment, we can try to make an environment which is as homogeneous as possible. Okay, so let's say we can. What we want to do is to make an environment that is as homogeneous as possible. The population can be also made simple by taking only a few individuals. In this way, we will limit the competition between individuals for the resources. This way, we will be able to avoid and to neglect the interaction between individuals. maybe we want to focus on small time scales to avoid mutations and to avoid possible trait evolutions. We would like to work on a controlled conditioned experiment because of repeatability. Okay, so we would like to make some experiments that are repeatable, and we would like to have some repetition of the ecological process and the ecological system, and we would like to make some, if it's possible, to make some direct observations from directly in the population. And if we take all this into account, if this ecological system could exist, then it could be well modelized by a simple linear burst dust model. Okay, so this was my idea. Okay, and okay, let's say, does this ecological system exist? And if yes, then we could be able to modelize the dynamics of this kind of population system with a very simple stochastic model. So of course, this kind of ecological system exists. So it's from a very recent paper by Jessica Coates and collaborators, published in 2018. So here I show you one of the pictures they have from the experiment. So they are able to take pictures of the population, and every little cylinder that you can see here in gray are one individual cell. And so they were able to count every individual cells in the population, and they can take some pictures of the population from time to time, and so they can have some very close to so they can have some observation for that are very close to the real dynamics of the population. Okay, so just to show you. Yeah, so what is this this experiment more precisely? Okay, so the protocol of this experiment is that they inoculate colonies of this bacteria of Escherichia coli on plates, and they inoculate every colonies with a single cell. So we know exactly the initial condition. We know that every colonies are inoculated with a single cell, and all single cells come from the same population, and so all the cells are can be considered as exactly identical. And so we have different colonies that are inoculated and initiated in almost exactly the same conditions. So they take a picture of the population every more or less 10 minutes, something like this. So they consider very rich medium. So there are plenty of resources to limit competition or to avoid competition. And of course, here we have a very rich medium and a very little number of single cells into the population. So the competition can certainly be neglected. So the protocol involves an automatized cell count. So they take regularly pictures of the system, and they have a software to analyze the pictures, and they can count automatically the cells into the medium. And what they did in the experiment was to look at the dynamics of the population of bacteria into 3 different mediums. The first medium is without any antibiotics, so it's antibiotics free medium, so it's a control it's a control population, and they wanted to compare 2 different kinds of antibiotics. The first one is what they call the bacteriostatic antibiotics, and this antibiotics is known to slow down or stop the bacteria growth and division. Okay, so if you put this kind of antibiotic, the bacteriostatic antibiotic, into a medium, then the bacteria does not die, but they tend to stop growing, or at least to slow down the growth. And the other, the third kind of medium is with another kind of antibiotic, which is a bactericidal antibiotics, which in this case does not affect the growth of the population, but it kills bacteria. Okay, so here we have three different medium with no antibiotics, with an antibiotics which slow down the growth rate and the birth rate and the division rate, and the third one which kills bacteria and so that increase the death rate of individual cells. So here, what they wanted to know is whether the dynamics are different between the three mediums. And here, what we want to know and what I propose to you today is by analyzing the dynamics, what we want to know is can we recover that the antibiotics that are used in these experiments differently affect the individual birth rate and the individual birth rate. Okay, so we suppose that the two antibiotics, the two kinds of antibiotics affect differently the individual birth rate and the individual death rate. Are we able to find this different effect of these two antibiotics from the population dynamics by inferring the birth rate and the death rate from the dynamics that is observed in the population and in the data? Just to show you, I have some video to show you the dynamics before showing you the data. So here, so you should see a picture of the two colonies of bacteria near the inoculation. As you can see here, we have a colony here with 2 bacteria and here with 4 bacteria. So we are very close to the initiation. Here, this is almost the first picture of the population. And so I will just show you a movie that shows you the dynamics. And in this case, it's without any antibiotics. So it's an antibiotic-free experiment. So it's the control population, okay? So there is, okay, you can see a growth which is very rapid, okay? So it takes some, maybe some, a few hours to have this kind of dynamic. So as you can see, without any antibiotics, then the population grows very, very rapidly and certainly something like exponentially. And as you can see, we have relatively good pictures here. And so we are able, at least until some point, to count each individual cell in the population, okay? So we can compare this kind of dynamics with a dynamic with the first kind of antibiotics, that is the antibiotics which is bacteriostatic, that limits the population growth, but which does not affect the individual death rate, okay? So here, this is basically the same thing. We have the same picture at the very beginning of the experiment, with here 4 different colonies that just started to divide. Here we have only 1 cell here, 1 here, and maybe 2 here that are beginning to divide, and 2 here. And we will have a look at how the population grow. So as you can see, this is the same speed, okay? So we can see that the growth is much slower, okay? But the bacteria divides and the population tend to grow, and tend to grow certainly something like exponentially, okay? But as you can see, the growth is very slower, is much slower than without the antibiotics, okay? And I will show you a last movie, and in this case, this is the last medium. This is the medium with the bactericidal antibiotics, which kills bacteria. Okay, so this is the same speed as before, and as you will see, there will be a very different dynamic. Okay, so as you can see, there are some division of bacteria, but some colonies went extinct. And some other not, and there is one population that managed finally to explode and to grow like something like exponentially, or these kind of things. Okay, so as you can see, there are here in this case with bactericidal antibiotics, then we can see that there are some extinctions, while other colonies avoid extinction and can grow exponentially. Okay, so this is basically what happens in the experiment. And now I will show you the data more precisely. Okay, so back to my presentation. If there is a problem with the presentation, just tell me, but I think it's okay. Okay, so what are the data? So the data we have are represented like this. I requested the data, the raw data from the author. So they were kind enough to give me the raw data. I just show you some figure representing the data here. So the first figure on the top left here is the population dynamics of the medium without any antibiotics. So this is the control experiment here on the top left. Okay, so what is represented here? It's basically it represented how the number of cells grows in number as time pass. Okay, so every as you can see, there are different independent trajectories here. So each point have a different color. Each dot represents one picture that has been taken automatically by in the population by the system. Okay, so we have each dot represents the number of cells observed in one colony at a given time. And we have a few dozens, and between 10 and 20 different trajectories for each colony. Okay, and as you can see here, there is no antibiotic in this medium here on the top left, and there is a very rapid exponential growth of the number of individuals in each colony. Okay, on the top right here, we have the same representation of the dynamics. We also have different trajectories. Okay, each dot also represents the number of cells in one colony at a given time. Okay, and here on the top right, it shows the population dynamics that has been observed in a medium with the bacteriostatic antibiotics, the antibiotics that slows down the growth rate and the division rate, but that does not affect the death rate. Okay, so as you can see, there is also here an exponential growth, but much slower than without any antibiotics here. And as you can see, there is some difference between the different colonies. Okay, so we have the trajectories are a bit different, even though the initial conditions are the same. So we have some variance in the trajectories of the different colonies, even though the experiment are exactly the same. The initial conditions are the same, and the environment are exactly the same. So we can see here that there is some stochastic processes behind these different population dynamics here. And on the bottom of the figures here are the population dynamics that have been observed with the third kind of medium, with the bactericidal antibiotic. Okay, so here basically on the left we have, on the bottom left we have the population dynamics that has been observed with bactericidal antibiotic, but with a low dose of antibiotics. And as you can see, then we have very different trajectories. Some trajectories go extinct and some others grow exponentially. Okay, and some others stay relatively stable, at least during this time. And on the bottom right here, this is the same antibiotics, but with a very high dose of antibiotics into the medium. So here, as we can see, all the trajectories go to extinction. Okay, so here the dose of antibiotics is so strong that all bacteria die before that the population grow exponentially. Okay, so here maybe we can interpret these two kind of dynamics with a low dose of antibiotic of bactericidal antibiotics on the left, and with a high dose of antibiotics on the right as a supercritical stochastic nonlinear process, birth-death process, and on the right, a subcritical linear birth-death model. Okay, so here, this is the data I acquired from the author. And the thing that I wanted to do is, okay, I have this data. Can I get the information about the birth rate and about the death rate from in this population, in these controlled experimental conditions? And I am able to show that the different antibiotics affect differently the birth rate and the death rate. So for doing that, I first applied a very simple model. So I made inference from a pure birth model. So it's called a Yule's process. So what is the model? So the model is the following. So what is followed is the number of cells at time t, so xt. So xt is a random variable that shows the number of cells in a colony at time t. The parameter of the model is the individual cell birth rate, okay, little b. And another parameter is given by, is controlled. It is given by the observation, and this is the time frame, delta t, between two observations. And this is given, the delta t is given by the experiment, because we know at what time each observations have been done. So a pure birth model can be formalized, and its distribution is known. It's a negative binomial, which is really known. So we know exactly what is the probability in a time frame delta t to have a variation in the size of the population of size small k, conditional on the size of the population n at time t. And this is exactly given by this this negative binomial distribution, with p here is the probability to have an event, a birth event, during the time delta t with a birth rate b. So since we know exactly the distribution of the number of cells at any given time under this stochastic process, then we can calculate the exact likelihood of the parameter b given the data. Okay, so we know exactly this, so we can write the exact likelihood. And here, where the observations are the number of cells observed at time I, in observation I, and ti is the time observation of the observation I. Okay, so basically what I did was to apply this likelihood on the data from this experiment. Okay, and what I wanted to do was to infer and to estimate the individual birth rate. So here are the results. So without any antibiotics, the estimated birth rate, so the b hat here, is 0.023. I forgot the scale here, but it's per minute. Okay, so the birth rate per minute is 0.023. I just give here the AKIK and the Bayesian information criterion. I don't use it now, but I will use it later. But I computed the AIC and the BIC. And with the bacteriostatic, with the antibiotic which slows down the birth rate, then the estimated birth rate was lower than without antibiotics, and was estimated at 0.014. It was not possible to estimate the birth rate for the two other cases with the bactericidal antibiotics, because there was some extension, and because there are some extension, then the likelihood is not defined, and at some point, and so it was not possible to define and to estimate the b hat under this model. Just for a comparison, in one of the author of the paper and of the experiment told me, I gave him my estimates, the estimation I had from the model, and I told him that, well, the birth rate I inferred from the data was, without any antibiotics, was 0.023, and he was quite surprised that, in fact, it really corresponded to what he estimated from direct observation. When they observe directly the rate at which one single cell divides into this kind of experiment, they obtain a cell division rate which is about 0.025 per minute, which is very close to what is estimated from the Yule's process model. Okay, but here with this model, I cannot estimate differently the birth and the death rates. So I wanted to apply and to make some inference with a different model, with a linear birth-death model. So here it's a bit different. The difference is that we have the same thing that before. That is, the xt is a random variable giving the number of cells at time t. Here we have also the cell birth rate, B, but we have a new parameter, D, which is a cell death rate. I introduced also gamma, which is an allometry parameter scaling, gamma, which is defined as B plus D, the sum between the birth and the death rate, which is an allometry parameter, which allows to scale at which rate all the demographic processes occur. I have a parameter here with the delta t, which is given once again by the experiment. So we know at what time all observations are done. And this model is not, there is no, at least as far as I know, there is no simple distribution that can describe all these processes. Then we need to make some simplification of this model. And to make some simplification of this model, we can make the assumption that the birth dates and the death rate and the population size scale with the parameter k, and we let this parameter k goes to infinity, which allows to get some approximation, which is shown by Vincent Bancet and Sylvain Billiard in their books, for example, that this linear birth death model can be approximated by a stochastic differential equation, which is like this. So the variation in the number of cells between two time, dxt, is equal to a deterministic part here, which is the difference, which is given between by the difference between the birth rate and the death rate multiplied by the size of the population and by the time frame we consider, and a stochastic part here, diffusion part, which depend whose variance depends on the allometric parameter gamma here, and which follows a Brownian motion. Okay, so here we have two informations. The first information comes from the mean trend of the trajectories, and the other information comes from the variance and the fluctuation between the trajectories. And what I have done is to use these two information to estimate separately the birth rate and the death rate. Okay, so for doing that, I discretized the stochastic differential equation using a Euler scheme. Okay, so this is basically this equation here. So it's possible to consider a delta t which is not so small, but which correspond to the time frame observed in the experiment. Okay, so basically this is the discretization of the stochastic differential equation shown before. And once I have this discretization, it's possible to calculate the probability of any change in the population size given the population size at the time t. And so it's possible to calculate the exact likelihood under this Euler scheme of the stochastic differential equation. So it's a bit complicated to make some inference of B and D with this likelihood approximation, and it has been done fortunately by Maud Delattre and collaborators. So they showed that in fact, the mode de Lattre showed that this approximation of the likelihood is a good approximation of the process, and they provided an R package which allows the estimation of the parameter of the birth-death model here by using this kind of trajectories. Okay, so I use basically the algorithm given by de Lattre and collaborators, and just a few details. So I introduced a mixed effect in the Druid part, so in the deterministic part. That means that I allow that there are some differences in the parameter between the different trajectories. It can occur or not. And I suppose that there is a fixed effect of the parameters in the diffusion part. So I have only one single parameter value for all trajectory in the diffusion part of the system. Okay, so I will give you the result I obtained. So here, without any antibiotics, I obtained an estimate of the birth rate like 0.039 and the death rate 0.016. With the bacteriostatic antibiotic, I estimated 0.016 as the birth rate and the death rate as 0.002. So here we can see that even though the experimenters think that the bacteriostatic do not affect the death rate, in fact, the model makes some inference that the death rate is not equal to 0. And in this case, even though I have some extension in the populations, then I can estimate the B and D. And okay, so here when the antibiotic is a bactericidal, I have a 0.023 birth rate and 0.017 death rate. And in the case where all the trajectories went extinct, then I estimated the birth and the death rate as the birth rate is lower, for example, here than the death rate. Okay, so as you can see, it's possible to use the deterministic trend of the trajectories in the population to estimate the growth rate B minus D, and it's also possible to use the variance and the fluctuations in the trajectories to estimate the variance of the Brownian motion, and since this variance is proportional to B plus D, then it's possible to estimate separately the birth and the death rate. So if I compare, and we show that there are some issues from the models and from the inference, and I don't know what to do with that, in fact. Okay, so just let's compare the result I get from the pure birth model on the first lines and from the birth death model on the second part of the table here, on the bottom part of the model. So here I just showed you a short reminder that what are the estimated value of B and D I obtained just before. And just to show you that when I take this estimated value of B and D, what is the growth rate? That is, what is the mean growth rate? What is the exponential growth rate of the population? That is basically B minus D. So if I calculate B minus D from the estimations, what do I obtain? Of course, when there is a pure birth model, the growth rate is basically equal to the birth rate. Okay, so I have it here. And when I compare the growth rate obtained when I subtract the birth rate and the death rate in the linear birth-death model, so as you can see, I exactly obtain the same growth rate. Okay, so here my question is, okay, why? What is the meaning of that? I obtain the exactly the exact same growth rate, and what is the true answer here? The true answer is effectively even in the control population, the individual cells die at some rate, or do the individual cells do not die in the control population? Because at the end, what is important is the net growth rate here. So what is the meaning of this death rate 0.016? Does it have any meaning? In fact, it is not clear, but when I apply the model, then it gives me some estimation. But if this estimation of the death rate is not correct, then The problem is why it should be correct for all the other estimation. So for the bactericidal antibiotics, I also obtained some estimate of the death rate, okay? But if I trust these estimations, why should I not trust these ones, okay? So here I have an issue is how could I say when or not the estimation of the birth and the death rate are correct, and it's not really clear for me. Okay, so here for me it's an issue. What is the good answer here? It's not clear. If I have only this kind of information, I cannot decide whether or not the death rate is the individual cells effectively die at a given death rate. There are also issues from the data.
Excuse me, Sylvain,
yes?
is it possible to conclude in a few minutes? Or?
Yeah, of course. Yeah, I'm sorry. I will conclude. Just to summarize, so there are some issues from the data because, in fact, it's I don't know if you see that very clearly, but in fact, the first observation that have been made in the experiment come after a few dozen minutes. And in fact, there is no observation between the inoculation of the population, the colonies, and the first observation after a few dozen minutes. And in fact, we don't know whether or not there are some colonies that were extinct during this first time frame here. And in fact, there is no reason that no colonies are not extinct during this time frame at the beginning of the experiment. So here we certainly have an observation bias because all the observation and measurements that have been done in the experiments are conditional on survival after a given time point. And so this is, I think, one of the issues from the data is that, in fact, the data are conditional on survival. And so this, in some point, should be included into the model and the inference. And how to do that, to me, is an open question. And just to finish, I think there are also, all that gives me some questions about the interpretation that we could make from the estimation. So as we could see into the movies, the population were clearly specialized, but is it important or not, it's not clear. Of course, since the population are specialized, there should be competition between individuals. And as we should see also, the medium is certainly not constant and certainly not homogeneous because there is a consumption of resources for antibiotics, okay? So what does mean exactly the estimation of B and the estimation of D hat? And to me, it's not clear what is the real meaning of B and D. So there are also questions of all that, but to me, the question is, even in this very simple model, this is maybe one of the simplest model and the simplest population biology system we could imagine and we could consider. Even in this case, it is not clear to me what could be the most relevant model to correctly estimate the birth rate and the death rate on such data. And to me, this remains an open question because, and I think it is important to answer this question because if we are not able to answer this question, even in this very simple model, in this very simple population biology system, then maybe we have very, very little hope on being able to do anything with much more complex systems. So I just would like to acknowledge and to thank the organizers, of course, and Min-Soo Kim for sharing the data that I used in this presentation, and Maud Delat for discussion and for help for the parameter estimation from the stochastic the differential equation. So thank you and sorry for being late.
Thank you very much, Sylvain. So I think we can go on with questions and continue with the discussion session right now. So do you have first specific questions for Sylvain? And then we carry on with maybe links between the two talks. Yes, Alex.
Hello. Thank you for this interesting talk. Can I just ask a very simple question? You mentioned the estimates of the paper authors of the growth rate. What do they mean by growth rate?
I think I'm not a microbiologist, but I think that what they did is that they are able to look at under the microscope, for example, to look at a single cell and to calculate and to estimate at which what is the time taken by one cell to divide. And I think this is the thing that they are able to do now, and that this is the way they estimated directly the birth rate. Yes, I think
so this is really independent of the data.
Yes, okay. I think so.
I was wondering if it was actually the effective, if it was B minus D, in fact.
No,
from the population size. But.
Yeah, I think it could be very interesting to think about what is exactly they estimate when they look at a single cell. Yeah. Is it exactly the same or not? Is it because, of course, this is conditionally on the survival of the cell and on this kind of thing. So maybe it's biased in some way. So I think it could be very interesting to think about what is the meaning of a direct observation of the birth rate and of an indirect estimation of the birth rate using this kind of data. I think this could be very interesting to think of. Yes, to think about.
We have some questions of Christophe Pouzat. How is the validity of the SDE model checked, and what do the standard errors of the estimated parameter look like?
I'm not sure to understand what you mean by the validity of this model. I think this may be one of the questions I had. I did not have the time to talk about that, but one of the as far as I understand the models, one of the hypotheses which is made in the linear burst model, for example, is that the time of events are exponentially distributed. And this is the thing that there is no reason that it works effectively in biological system. So maybe this is one thing that could be checked, but I don't know how it could be done. But with this kind of data, is it possible to check whether or not the time of events are effectively exponentially distributed? I think this could be a thing that we could do to check whether or not this kind of stochastic process correctly modelize the biological process. And maybe the question is about the approximation, the validity of the approximation, right?
Yes. So, and I agree with you that you can and should question the fact that you have exponential life distributions, but then you go one step further when you go to the stochastic differential equation model, and underlying that you're assuming you have a large population and you apply that to tiny ones. So another way to ask the question is, if you take the parameters you've estimated with the SDE and then simulate many realizations, where does the actual data set, where is it located compared to the population of simulated data?
Yeah, I agree with that. I don't know. It's a good question. I think it's really important to do to check that, in particular because in the experiment we observe extinctions, and extinctions are not rare at all. So of course, if there are extinction, populations are not large, of course, and so it's a very good question, and I don't know, and maybe it's and I really don't know. I don't know. And I even don't know how to do that. We could do that by simulation, of course. But I did not do that. But maybe because it's a very simple model, maybe it could be we could do something much more mathematically, and more formally than by doing that by simulation. I hope so. But how to do that? For me, it's an open question. And about the standard errors, so I did not show you that, but I calculated the standard errors by making some assumption on the distribution of the parameters. Basically, I suppose that the parameters are normally distributed, basically, and the standard errors are very small. So I think there is no problem from this point of view. I think there is no problem about the variance of the estimation as far as I did the thing correctly.
Maybe I just have something. I saw that Patrick raised his hand, but I just have something very precisely on this question of individual cell measurements. In fact, there are teams now who measure individual properties of single cells, right?
Yeah.
So in fact, you could test if the distribution of the death are exponential, for example.
Yeah.
I guess so. It wouldn't be the same experimental setup, but
yeah,
I guess you could cook up some experiment for these new labs,
yeah,
where they can.
Yeah, but to me, you know, if we need to look at individuals' fate and division rate and so on to make some inference about their rates of birth and so on, if we have to look at and to observe individual cells, so what is the use and the meaning and the need to make some inference from population dynamics models? Maybe we don't need to do that, because if we cannot be sure that our estimation are good from the population dynamics, because we need some direct observation to be sure, though maybe it's not a good idea to make some inference from population models. I don't know if you see what I mean.
I'm not sure. So I guess there are microscopic behaviors, like
yeah.
microscopic laws
yeah.
or mechanisms, like birth, death, switch, or whatever. And there is a macroscopic level, which is the kind of scaling, like in statistical mechanics. And if you have right estimations of the mechanisms, the biological mechanisms, then it makes sense somehow of doing the scaling.
Yeah,
there is a canonical one.
Yeah, I agree with you. In fact, this is the thing that I wanted to discuss also, is that in fact, maybe the question which is not clear to me is what is the scale of observation, in fact, and maybe when we measure